All right, everybody. Well, I think it's time for us to start. I'd just like to quickly say welcome to everybody. And this is going to be an interesting experience to just start everything off. So I'd just like to welcome everybody here today. And the first thing my name is Sally Turnbull, and I'm the Administrative Officer at Retina Australia. And I'll be sort of administrating this webinar today. And so this is our very first webinar, and there may be a few technical things that happen, which we hope that won't cause a problem for you. So what I want to do now is introduce Leighton Boyd, the chairman of Retina Australia, to just say a few words. One thing I would say, we've had a query from one of the attendees. All the attendees, you can't speak. You've got your microphones muted and you won't be able to speak during this meeting. But you will be able to type questions and we will be able to answer them that way. So that's the way it works. Don't panic if your microphone doesn't work. That's how it's meant to be. All right, so let me see if I can introduce Leighton. Just a moment. Okay, Leighton, you're ready to go.
Thank you, Sally. Look, I'd just like, on behalf of the Board of Retina Australia, to welcome you all to our webinar. Certainly, COVID-19 has affected all of us, both here in Australia and across the world, as you are all well aware. Technology is an amazing thing, and as far as us in Retina Australia are concerned, the use of webinars have never happened, as in the organising of it, and a number of us during this difficult time would have gone to Zoom meetings and webinars and various other technological things. We're very lucky today to have 4 presenters who will talk to us. It's also probably a first time for them. So I want you all to enjoy the next hour or so. Give us feedback and just take in the moment. It's thanks to Sally. Sally has done so much research on this matter and has put together things, and with the support of Fake in the office and June. So thank you, Sally, for all your work. I'll hand back to you.
Okay, so
Thank you.
All right, so our first speaker this afternoon is John De Roach. Dr. John De Roach is the Principal Medical Physicist for St Charles Gardner Hospital, an Adjunct Associate Professor in the Centre for Ophthalmology and Visual Science, University of Western Australia, and is the Principal Investigator for the Australian Inherited Retinal Disease Registry and DNA Bank. John is one of 4 speakers today who was instrumental in creating the IRD Registry and DNA Bank in 2009. He is responsible for the management of the registry, including the attracting of research funding to sustain the registry. Today, he will introduce the webinar with a brief summary of the registry and DNA bank. So now I'm just going to hand over to John and hope that he's got everything that he needs.
Thanks, Sally. Okay, can you see and hear me, Sally? She's muted. Okay, well, welcome to this webinar. It's a bit of an experiment. So today we have 4 speakers, myself, John De Roach, Terri, McLaren and Tina Lamey. We're all from the Australian Inherited Retinal Disease Registry and DNA Bank. And Rachel Heath-Jeffrey, who's from Lions Eye Institute, which is an institute with which we collaborate very closely. And a lot of you will be aware of Fred Chen and David Mackey, who are part of that institute. And the 4 of us will be speaking in turn. I'll be speaking about the Australian Inherited Retinal Disease Registry and DNA Bank, just to give a bit of an overview. Terri will speak about genetics reports we provide to some of our participants, which is often of great interest to them. Tina will give an overview of the various therapeutic strategies and clinical trials and treatments that are happening around the world. And then Rachel will give an example of a specific study of one of those, or a presentation of one of those specific studies being carried out at Lions Eye Institute at the moment, called the Western Australian Retinal Degeneration Study, as a particular example of what Tina will be talking about. So we expect to speak for about 45 minutes, and after that, we're happy to answer any questions. So, hasn't changed on here, but anyway. So I'll now speak on the Australian Inherited Retinal Disease Registry and DNA Bank. So inherited retinal disease is the major cause of blindness in working age Australians. It used to be diabetic retinopathy, but that's been brought under control very well now, medically. It can be caused by any one of more than 260 genes, mutations in those genes. It's had many demonstrations of disease arrest or reversal in animal models. It's a subject of many promising human clinical treatment trials around the world, but mainly it requires definitive genetic diagnosis in order to be appropriately managed and ultimately successfully treated. It's not funded at present, or never has been, by Medicare, the genetic diagnosis of inherited retinal disease. And because of that, in 2009, Retina Australia provided funding to establish the Australian Inherited Retinal Disease Registry and DNA Bank. We're just gonna look at the technical issue we have here. So, sorry. And this is now a mature national research resort situated at Sir Charles Gardner Hospital in Perth. Sir Charles Gardner Hospital is a large 600-bed teaching hospital. It's very, it's a hospital that's been here for a long time, or unless Clive Palmer has his way. And it's the resources are well organised, enduring repository of demographic, pedigree, genetic and clinical information from Australians affected with an inherited retinal disease and their family members. So it's a registry and a DNA bank. It includes DNA and participants' IRD causing genetic variants where we have established them. And I'd like to emphasise it's a research tool, it's not a clinical service, although genetic results are reported to participants where possible, when we can, and that's what Terri will be talking about in the next part of the talk. So this is a pie chart, for those who can't see it, of all the diagnoses captured in the registry. It's notable that we don't look at age-related macular degeneration. Almost half are retinitis pigmentosa, and I'll just go through the others in decreasing order of frequency. Stargardt disease, cone and code-rod disease, macular disease, Usher syndrome, Best disease, retinoschisis, Leber's congenital amaurosis, Bardet-Biedl, choroideremia, and then there's other, which makes up something like an eighth of the whole chart. So they're the main diagnoses that we deal with, and we have research programs going for most of those with various collaborators. So a little bit of background about how we recruit people and so on. When we're recruiting participants, we typically obtain information by telephone because we recruit from all over Australia. And once someone's interested in being a participant in our registry, we obtain written consent from them to participate, to collect their DNA, and also to request relevant clinical information from other clinics and institutions that they may have attended, because we're not actually a clinical group, although we have a lot of clinical people that we collaborate with, but any clinical information we have to get from participants, doctors, ophthalmologists, and so on. The information in family trees are stored in an electronic database, but not all information is stored electronically, and we also have now 20 filing cabinets full of information. Here, for those who can't see, this is actually 2 photographs of all of our filing cabinets being displayed by Ling Hoffman, who's sitting in the audience here. And Ling is one of our recruitment people, and the filing cabinets, for those who can see, actually go way past filing in the right-hand slide. So we have 20 filing cabinets. We didn't know where to put them. They're all in the corridor, all under lock and key, of course, but which are pretty much full now, containing all the information we have of all the people in our registry. So that's sort of a tangible picture of our registry. But a lot of the information is, of course, also stored electronically, so we can query it and so on. In terms of DNA collection, we collect blood or saliva. We prefer blood because we can get a lot more DNA from the blood than we can from saliva. We collect it Australia-wide and internationally. We're an Australian resource, but a lot of people in Australia, of course, have relatives who live overseas, and often we need their DNA as well in order to carry out a genetic analysis for the family. And participants are asked to visit the local pathology centre with a kit that we send them, and the pathology centre then collect the blood and ship it to us, and the DNA is then extracted and is stored in a DNA bank which is situated at the Lions Eye Institute. And there's no cost to the participant in that except getting themselves to and from the path centre, of course. This is my last slide, actually. It's a bit of a busy one, but just to give you some numbers, to give you an idea of the scope of the registry. The DNA bank contains DNA from more than 7,300 participants. 60% of those are affected or carriers, and the remainder are family members, blood family members, who are either unaffected or unknown, sourced from over 2,000 families. And we estimate we have DNA from about 25% of all IRD affected people in Australia. So that it's quite a chunk. Genetic analyses have been performed for more than 2,600 participants, which is, if you'll note, is only about a third of all our participants. And approximately 500 families have had their genetic cause established. And the intention is to genetically diagnose all families. So there's two messages here. One is we've done an enormous amount of work because a lot of the genetic analysis costs thousands of dollars for each one, and we've done more than 2,600, and we've diagnosed 500 families. Having said all that, we're only about a third or a quarter of the way through all of our DNA. So if you ring us and say, have we looked at your DNA yet? There's about a 1 in 3 chance that will have happened. But the DNA is sitting in a DNA bank, which will be viable for many years, and we're working through it. So in addition to that, as a sort of an outcome, when we do analyse DNA and we diagnose a family, we provide a research genetics report to a person's ophthalmologist or clinical geneticist if they would like us to do that. And that's what Terri will talk about in a minute. And we've provided more than 1,000 reports. So even though we've got a long way to go, we've actually done a lot as well. And in addition to those reports, we've got a lot of research projects underway focusing on many of the diagnoses in the registry. And in the past 3 years, as a measure of that, if you like, we've published or submitted about 30 papers to the peer-reviewed literature reporting results of our research in conjunction or in collaboration with other people, including and especially Lions Eye Institute. And if you want to know more about that, our annual report is available on our website. And I haven't got the website there. It's a great big long thing, but best is to Google it or ask us or Retina Australia. So that's the end of my talk. The next speaker will be Terri McLaren, and she will be reporting on the genetic results, the reporting of genetic results to participants of our registry via their ophthalmologists.
Okay, although you couldn't see Don's slide, my first one is the busy one. I'll just start by giving some background about the genetic results that we report to participants, and the results come from the research that we do. So in 2009, when the research, when the registry began, the research aims funded by Retina Australia were to characterise the genetic spectrum of inherited retinal disease, or IRD, in the Australian population, and establish and maintain a resource for researchers to use to develop treatments for IRDs. So reporting of individual genetic results was not a focus. Reporting of results is not a research activity, but a clinical service activity for which we weren't funded, and we haven't been funded to perform until very recently. Despite not being funded, we have provided many reports of results where the genetic diagnosis was likely to be of direct benefit to the clinical management of a participant's inherited retinal disease. In the years since 2009, the value and demand for individual genetic diagnosis has greatly increased because of advances in genetic knowledge, the improved availability of reliable genetic analysis, the increasing number of genes covered by clinical trials, and development of treatments, particularly personalised treatments. Okay, the next slide is a graph that shows the escalating rate of the number of reports that has been produced in the lifetime of the registry. So in the 5 years, 2009 to 2014, 215 reports were issued. In the 6 years since 2014, we've issued over 800 reports. So in total, we've issued 1,037 genetic reports for 704 participants affected with an IRD. So 678, or 96% of these reports indicated that a probably causative gene had been found. Okay, with this table, it shows the most frequently reported diagnoses. So they're retinitis pigmentosa, Stargardt disease, Usher syndrome, cone rod dystrophy. And this reflects the most frequently occurring diagnoses in the registry. The table also showed that some of the diagnoses are overrepresented in our reports, such as Stargardt disease. 22% of reports were for Stargardt disease, but that diagnosis is represented 9% in the registry. Leber congenital amaurosis, 4% reported, 1% presence in the registry, and similarly choroideremia, 4% reported, 1% in the registry. But this reflects our most analysed cohorts, all of which have advanced gene or mutation-specific therapies well advanced in development. Some diagnoses are underrepresented, and the most in that category is Best disease, where it's subject to 2% of reports, but 5% of the registry. And the table also showed that we have over 3,400 unreported affected participants. So, as John said, we do have a long way to go. So these have either not been analysed or they're part of the way through the analysis and reporting pipeline. So that brings us to the current situation, so reporting in 2020. While the registry remains a research tool and not a clinical service, we continue to proactively report where we believe it will make a difference to the management of the participants' IRD. And our capacity to report has greatly improved this year as one of our team members, Jen Thompson, received a grant from Retina Australia for 2020 to issue 200 genetic analysis reports. And just to put that in context, the registry recruits about 500 new participants per year, including family members. So while 200 reports is good, it doesn't meet the growth in the registry or the backlog of results that we have. So consequently, reports have to be prioritised. So the priorities for reporting a genetic diagnosis, if we find something unexpected, then we do report to clinicians so that clinical evaluation can be done for the affected person. So sometimes we're expecting a gene that's causing an IRD, but in fact we find a gene that causes not only the IRD but other organs or systems of the body. And similarly, a molecular diagnosis that's different to the clinical diagnosis might have implications for severity and management of the condition. We report if the causative gene or mutation found is a subject in clinical trials or developing treatments, and that is covering an increasing number. We receive a lot of requests from participants or their ophthalmologists or their clinical genetics service. And as John said, we try to meet that as best we can. But there is a one in chance that if you call us and ask that, that the analysis hasn't been done. So ultimately, we aim to establish the causative gene for all IRD affected participants and to report these results to the nominated ophthalmologist or clinical genetics service. Besides the main priorities, a genetic diagnosis is important for genetic confirmation of a clinical diagnosis for carrier status, facilitating personalised treatments, genetic counselling for family planning purposes. So just some practical things to know about reporting of results to participants. We only report genetic results when we have obtained a participant's specific consent to do so. If we have a result to report, we contact the participant and ask them to complete a consent to release information form, or CTR, where they nominate the ophthalmologist or clinical genetics service whom they wish to receive their results. We aren't able to provide results directly to registry participants, as is prevented by our research ethics protocols. We're a scientific research group, and results that really need to be given by a medical or genetics professional so that the consequences can be managed. And once the results have been sent to the nominated specialist, we inform the participant so follow-up can be arranged. And there are a number of steps we have to go through before results can be reported. We start by analysing the DNA of one IRD affected person in the family. Analysis takes a turnaround of can be months. When we receive the results back, they're assessed for validity because the lab might specify their findings, but our genetic scientists interpret the results and relate it to the clinical diagnosis and more knowledge that they have. We also might have to perform additional analyses. We don't find the answer in the first pass, but specific other tests may have to be used. Then we also have to confirm that the gene mutation found is indeed the one causing the IRD by analysing the DNA of parents or other family members. So that's another round of analysis, but takes a few months. And then we have to assess the specific gene mutation for pathogenicity. This is a complex and time-consuming activity that's carried out by our genetic scientists, Jen Thompson and Tina Lamey. So, and the reasons why a report may not have been done or is not available, we might not have performed the analysis yet, and that's really a funding and time issue. The analysis may still be underway because there can be many steps. The analysis may have been done, but results are inconclusive or negative. Current technology doesn't cover all the answers or find all the answers, or it might be that we have to go to other technologies like whole genome sequencing, which is expensive, not as readily available, and quite difficult to interpret. The results could be on a wait list for pathogenicity assessment, and we could have gone through all that, but the results are still on a wait list to be reported. It's the second last slide now. This is just an example of a report. It's generally about 1 to 2 pages. It states that it's a genetic analysis research report. We cover usual demographics, purpose of the test, why the analysis was done. We have a table of genetic results showing the gene causing the inherited retinal disease, that's if one is found, the specific mutation or variant in the gene causing the disease, pathogenicity, like it's pathogenic, likely pathogenic, variant of uncertain significance. We document the inheritance mode. There's a description of results where all the evidence is brought together to explain why we think that is the cause and how likely it is the cause of the diagnosis. Then the conclusion, is the clinical diagnosis confirmed or not, and recommendations maybe for further tests or actions. We cover the analysis methods, references used, and authorizations. Every report has a disclaimer where we state that research, the analysis is done in a research context and not a clinical context, and results are always recommended to be confirmed in an accredited diagnostic laboratory. Now, our results are extremely accurate. It's just because we're a research entity where we use a DNA bank, if results have to be confirmed, say for a clinical trial for pre-implantation diagnosis for family planning, then an accredited laboratory needs to be used. And so you use a fresh DNA sample and target sequence results that we have already found, but it needs to go through that step. So just in summary, it provides reports. I think I might have said this quite a few times, in a research context, not as a clinical service. Reports prioritise according to the benefit to management of participants' IRD. Reports depend on genetic analysis being completed, assessment of results and pathogenicity, and participant consent. Resources permitting, the registry aims to sustain providing at least 200 reports a year. But ultimately, we aim to establish a course routine for all IRD affected participants and to report these results to their nominated specialist. Thank you.
And our next speaker is going to be Dr. Lamey. Tina is a senior research scientist at the Charles Gardner Hospital, an adjunct senior research fellow at the Centre for Ophthalmology and Visual Science, University of WA, and is an associate investigator with the Australian Inherited Retinal Disease Registry and DNA Bank. She was also instrumental in creating the IRD Registry and DNA Bank in 2009. She is responsible for genetic analyses carried out on the DNA of participants and for the interpretation of the results of those analyses. Today, Tina will give an overview of the trials and treatments available to people affected with an inherited retinal disease.
What I would like to talk to you today is a little bit about the genetic basis of inherited retinal disease and how this results in vision loss. What's happening in terms of the development of different therapeutic strategies for inherited retinal disease, clinical trials that are underway, and what we're doing at the AIRDR in this space. Nope. Right. Okay, so there's lots of different forms of inherited retinal disease, and for most of these, the photoreceptor cells, the rods and the cones, are the primary cell types affected. So which cell type is primarily affected really depends on the disease-causing gene. So just to go back for a second, most of you are probably aware that our rods are responsible for our night vision and our peripheral vision, and our cones are really important for our day vision and our functional vision, our important central vision. Okay, so as I said before, which cell type is primarily affected, whether it be a rod or a cone, really depends on the disease-causing gene. And this is because specific sets of genes tend to be important in the development and or the maintenance of specific types of cells in the retina. So for example, there could be 20 genes important for the development and maintenance. There isn't, but the development and maintenance of rod photoreceptor cells. So a problem in one of these is going to cause a problem in the rods and so forth. So this explains how mutations in different genes can result in diseases with similar characteristics. So an example of this is that we have 65 different genes that could cause RP. So a mutation in any one of these genes can bring about similar functional loss, so a rod cone dystrophy, for example. So these mutations result in cell dysfunction and death, and these affect the visual pathway. So really, the aim of inherited retinal disease therapy is to, in one way or another, preserve or restore this visual pathway. So, and the visual pathway is a process by which there's cells in our wafer-thin retina capture light, translate this message, I suppose, or this signal into a message of another form, and then transmit this to the brain. So what happens is that photoreceptor cells, which comprise the layer at the back of the retina, they capture the light, they transmit a message to the body, and this is a really simplified form of the visual pathway. I have to just disclose because I've got 3 ophthalmologists sitting behind me. But these photoreceptor cells capture the light, translate the message, pass it to the bipolar cells in front, and the bipolar cells pass these on again to ganglion cells in the front layer of the retina. There are a few more cell types involved, but we'll keep it simple. These ganglion cells are really long and they extend from wherever they are positioned in the retina through the hole in the back of the eye, and they bundle together to form the optic nerve and plug directly into the brain. So you should be able to see now that a problem with any one of these cells in this process due to a genetic fault could and does disrupt this process. And more often than not, it's the photoreceptors that are affected. So if photoreceptors at certain positions in the retina stop functioning and die, this means that we can't interpret vision mapping to these regions in the retina where the cells have died. So in this way, a loss of peripheral rod photoreceptors would be expected to cause a problem with peripheral vision, or a loss of cone photoreceptors at the macula would be expected to compromise central vision. So with all of this in mind, with what I've just described, what might some of the options be in terms of therapies? So, and this talk is a little bit longer than it should be, so I'm sorry about that. So what might some of these options be in terms of therapies? So we could fix the gene that's faulty, causing photoreceptor cells to stop working and die. So these are gene-based therapies. We could provide conditions that are supportive for the survival of photoreceptor cells. I won't go into detail. So these are neuroprotective therapies. We could supply the retina with new photoreceptor cells. So these are cell therapies, and this can be from supplying, like providing new cells to, implanting new cells, I suppose, or delivering new cells, or converting other surviving neurons with genetic programming into photoreceptor cells, or converting some of them so that they can then capture the light. So a couple of other options include providing alternative pathways of capturing light. So this could be giving retinal cells an additional capacity to capture light, so they maintain their role as well as have a capacity to capture light. So this is another emerging treatment strategy. And finally, one you would also be familiar with, I'm sure, would be the insertion of an artificial source to capture light and pass this message on. So this is retinal implants. Okay, so it's important to know that the application of therapies really depends on a couple of things. Also, the application of different treatment strategies would really depend on a couple of important things. So the first is knowledge of the genetic cause. Okay, so where a genetic cause is known, obviously then a gene therapy could be an option. Gene therapy really aims to address the root cause of the disease and applied at early stages nips the disease in the bud, essentially. This is the idea. And there are a couple of different strategies, and again, I won't go into a lot of detail, but I'll just briefly kind of describe them. So the first one that you're probably familiar with is gene replacement, and this is associated with basically replacing the faulty gene. So a good copy of the gene is inserted or is delivered to the retina, it gets incorporated in the photoreceptors, and then it can start working, doing its job, the cell's happy, hopefully functions, and doesn't die. So there are a couple of newer strategies that are emerging, and these either repair, I'm trying not to give too much detail, so these either repair, mask, or cut out a mutation. So it might seem counterintuitive that a mutation would cause a disease, but we can cut out a region of the gene containing the mutation and not cause a problem, but that is the case for some regions of some genes. So the latter two, these are more personalised because they are specific to the mutation and not just the gene. And one thing that I really would like to impart in terms of gene therapies is that these modifications can't be inherited. they're made, or these changes are made to retinal cells, not the gametes, not embryos. So if something goes wrong, that problem isn't going to be inherited by children, which is important. But it also means that children will have to have therapy if they also have the disease. Okay. So we know that knowledge of the genetic cause is a factor affecting the application of different strategies. But another is the stage that one is at in the disease course. So we know that as inherited retinal diseases progress, photoreceptor cells die. So if someone is at a stage of disease where they don't have any photoreceptors, we can't apply gene therapy because there are no photoreceptors to apply the gene therapy to. So in this case, there are various other options: neuroprotection of surviving cones, if, for example, the rods are primarily affected. There could be cell therapy, optogenetics, retinal prostheses, and whatever else might be emerging in terms of strategies. So a third factor affecting the application of any therapeutic strategy really is knowledge of how the disease is progressing in a given individual. And so this is important because you can't assess the efficacy of a trial on a moving target if you're not aware of how and the direction that that target's moving in, if that makes sense. So, but in any case, really what I wanted to show here is that there are lots of emerging treatment options that could be undertaken at various stages of the disease, but there's a few factors that we need to consider. Okay. So, given the importance of identifying the underlying genetic defect causing inherited retinal diseases, core projects at the AIRDR tend to focus on diagnoses for which gene therapies are emerging. So these are undertaken to a greater or lesser extent in collaboration with other groups locally, nationally and internationally. Okay, so really the role of the AIRDR in the context of developing therapies is the building of genetic knowledge. So John might have previously mentioned something like this, but we are deciphering and building a database of the underlying genetic faults in the Australian population. And this is guiding the development of therapeutic interventions for IRD-affected Australians, and it's facilitating the recruitment of candidates for current and pending Australian-based trials and treatments. And this is obviously really important. So these include candidates for a few recently approved trials. There's an antioxidant therapy, which is like a neuroprotection strategy for preserving cones in RP-associated Usher syndrome. There's a drug therapy associated with ABCA4 Stargardt disease, and you could talk to Fred Chen about this if you wanted more information. And of course, the RP65 gene therapy for Leber's congenital amaurosis, and I'll talk a little bit about that now. So, what's the status of various treatment options out there currently for inherited retinal disease? You'd have to be living under a rock if you didn't know that the first person was recently treated with an FDA-approved gene therapy for Leber's congenital amaurosis, and this is called Luxturna. So the aim of this is to replace a gene that causes one form of LCA. So, and a month ago, Luxturna was given TGA approval for use in Australia. So this is an Australian first. So through our LCA study, we've identified 4 possible candidates for therapy. And when I say possible candidates, I mean we've identified individuals with mutations in this gene. So there are various other criteria that have to be met, but they're candidates in terms of their genetic mutation or gene that's gone wrong. So in addition, because it's a pretty hot kind of topic at the moment, we've also recently provided 40 DNA samples to collaborators at Royal Victorian Eye, and they're testing these in the hope of finding more trials or more trial candidates. And our research collaborators in the Netherlands have also offered to analyse about, as it turns out, 550 to 650 of our autosomal recessive RP samples, and this, we hope, is going to identify more potential candidates for this therapy in our population. And this is actually really fantastic because we haven't looked at our autosomal recessive RP cohort as a core project yet. It's really big, so this is a fantastic way to get this analysed. So none of this would be possible without this Retina Australia funded registry having been established. Okay, so in addition to this, it's really important, I suppose, to let you know that there are about 20 more of these gene-based treatments in the pipeline, or sorry, gene-based clinical trials in the pipeline currently. So these are at, I think I've missed a couple here, but you get the idea. They're at various stages of completion. So most of them are in phase 1, a couple are in phase 2, and of course the RP65 Sanofi Luxturna is post-approval. So since these, sorry, I just wanted to check something. Okay, so these therapies are really aimed at addressing a number of different diagnoses. So this includes chromatopsia, chronic dystrophy, choroideremia, Leber's congenital amaurosis, retinitis, some forms of recessive RP, X-linked RP, Stargardt, and Usher1 and Usher2. But since these therapies are gene and sometimes even mutation specific, they're not applicable to all patients with these diagnoses. So that's something that's important to remember, with the exception of choroideremia and retinitis, because there's only one gene for each of those implicated in disease. And these genes are being, or therapies addressing gene replacement for these are in trial. So the gene therapy for chromatopsia, for example, there are 2 trials targeting or looking at gene replacement for CNGA3 and another 2 trials for CNGB3. And together, these genes cause about 70–80% of the chromatopsia cases. So we've identified about 35 registry participants with CNGA3 or CNGB3 disease through one of our core projects, which was on chromatopsia. So just to mention a couple more associated with some of our other core projects, we've identified mutations or participants with mutations in genes that are targeted in other trials. So we have 85 participants, or males actually, there are more females, but 85 males with X-linked RP due to mutations in RPGR. Now, it's important to reiterate that not all of these are going to be candidates for trials for a number of reasons, but we have this many individuals with mutations in this gene being trialled. In fact, there are 3 gene therapy trials running at the moment for RPGR. And this gene causes a huge proportion of X-linked RP cases. We have identified 250 participants with Stargardt disease due to mutations in ABCA4, 38 with mutations in the RS1 gene that causes retinitis, and 35 participants with mutation, did I say 35? Anyway, 35 participants with mutations in the CHM gene causing choroideremia, and there are lots more. So I think I've only got one more slide, or maybe 2 more slides left. So, but in addition to these 20 gene-based clinical trials, there are there's another whole set of clinical trials associated with non-gene-based methods. And all of this, again, is just the tip of the iceberg with respect to the amount of work that's going on developing and testing therapeutic options, I suppose, in cells, tissues, and animal models. So we should be expecting a lot more, and these are all geared towards, of course, the development of inherited retinal disease treatments. Okay. So the take-home message here really is that there are many different therapeutic interventions in development, but there's not a one-size-fits-all approach currently. And genetic characterization of our population is important. Obviously, we're doing this at the ARDR. So suitability for a given strategy really needs to be assessed on a case-by-case basis. And this will happen by asking questions like, what is the genetic basis of the disease, which is where we come in, and of course, clinical genetic testing. And what is the natural history of disease in a given case? So this is where you need to consult your retinal specialist for routine monitoring and natural history studies if they're available. So I will leave it here. But before I go, and before I hand you over to Rachel, actually, Dr. Lauren Ayton has asked us to inform you that at the Vision Optimisation Lab, they're running a natural history study for inherited retinal diseases that are at this stage closest to trial phase. And at the moment, they're actively recruiting through CERA, University of Melbourne, University of Tasmania, and Lions Eye Institute. So my apologies, Lauren, if I've got some of that wrong. Their group email address is up here on the slide, but you could also contact Retina Australia. So thank you for your attention, but don't go anywhere because Rachel is going to talk about the ward study now.
Our final speaker for today is Dr. Rachel Heath-Jeffrey. Rachel is the Miosevich Medical Retina Fellow for Dr. Fred Chen at the Lions Eye Institute. She is responsible for recruitment in the Western Australian Retinal Degeneration, the ward study, and facilitates the determination of the clinical phenotype. Today, Rachel will give an overview of the ward study available to people in Western Australia affected by an inherited retinal disease.
Hello, so my name is Rachel and I'm currently working with Dr. Fred Chen at the Lions Eye Institute. And today I'd like to talk to you about the Western Australian Retinal Degeneration Study, also known as the ward study. So the ward study was established in 2015 in December, and so it's been running for about 4.5 years. And this study, the aims are to 1, measure disease progression, and 2, to use patient cells in order to develop personalised drug therapy. We've received significant funding support from multiple sources that's listed. So in detail, the study is composed of 2 parts. The first part is to monitor the retina through detailed imaging every 6 months over a 5-year period, and so that we can measure disease progression rates. The second part involves the collection of a small skin and blood sample so that we can create stem cells in the laboratory, and that we can then use these to make a miniature retina in a petri dish so that we can understand how particular genetic mutations lead to damage at the cellular level. So this is an example on the slide of a patient that has been monitored with Stargardt's disease, and this patient has been monitored every 6 months over a 5-year period. The slide shows that there's a dark patch that's increasing in size over the course of the study. There's also evidence of flecks that are increasing in length throughout the study. And having close follow-up with detailed retinal imaging is important so that we can measure the disease progression rate, so that when we develop a treatment for this patient, we know the baseline with which we have a future comparison for. So this is another example on this slide of a patient that has retinitis pigmentosa, and this patient has been monitored very frequently over multiple years. There's an area in the central macula that's a bright kind of white oval region, which represents the area of the retina that is functioning well. And what it shows is through sequential imaging over time, this area is decreasing in size. So the ward study calculated that the rate of progression is approximately 10% per year. When we develop a treatment for this patient, we would expect that the change in this progression rate may go from, say, 10% to 5% per year. And if we don't have natural history studies, we won't know what to aim for. It's important to note that even patients with the same genetic mutation can have different rates of progression, and even the same patient between their right and the left eye can vary. So natural history studies are now widely available around the world in most developed countries. And the ward study is available to patients living in WA, but ideally this type of study should be available to patients living in all Australian states and territories. This slide shows on the right that there's a panel of a patient with RPE65 mutations. This can lead to retinitis pigmentosa or Leber's congenital amaurosis. And this patient has been monitored over a 6-year period. Studies like this were essential for the development of the Luxturna gene therapy. So part 2 of the ward study involves the collection of a small skin and blood sample so that we can create induced pluripotent stem cells in the laboratory. These stem cells are very useful because we can test new drugs on patients' skin cells first. We can also look to make sure that there's no toxicity effects and that there's reversal of cell damage. On the right, this shows 4 photos. So 2 are from a patient with CLN3 mutations and 2 are from a healthy control. On the upper panels, it's using a specialized microscopy technique known as phase contrast, and on the bottom panels, it's using fluorescence microscopy. And it shows that in the skin cells of this patient with the CLN3 mutations, they have abnormal accumulation of vacuoles. So these skin cells are useful because, again, we can test drug therapies on them, and they're now an FDA-approved platform for modelling the disease. So these induced pluripotent stem cells can then be reprogrammed to become a retinal organoid. Retinal organoids can then be used for testing, also drug testing, but also for modelling the disease and checking that how a particular mutation leads to cell damage. Retinal organoids, they're useful because some retinal genes are not expressed in the skin cells, so we need to have a retinal organoid in order to express the or activate the retinal genes. And this is an example of the developmental course of a retinal organoid of a patient with PDE6B mutations compared to a normal control from day 28 to day 180. And so what we can see is that it can be difficult to tell what cells are present, and therefore we need to use specific cell markers to identify which cells are there. So this is another slide, and it shows the maturation of a retinal organoid from day 45 to day 120. And you can, it shows that a variety of cells are present, and in order to identify these cells, we need to use very specific cell markers. So what have we achieved in the ward study? Well, to date, we have over 440 patients that we have enrolled, and of these, 2/3 have a genetic diagnosis, of which this has been provided either by the AIRDR or Genetic Services WA. Of our patients, we've found that 62 different retinal genes have been implicated, of which the most commonly affected are ABCA4, which causes Stargardt disease. The second most common was PRPF31, which causes dominant RP, followed by RPGR and USH2A. So this is a timeline that shows our progress in the ward study. So as I mentioned, it was started in 2015, and 6 months later, a research collaboration was established between the Lions Eye Institute and Murdoch University in order to develop a treatment, a drug treatment for PRPF31 or RP11. So 3 years later, this novel drug has attracted attention from PYC Therapeutics, and so another collaboration has been established. Over the last 12 months, we've been further refining the drug and on patients' skin cells, and also identifying more patients with this particular mutation from the IRD registry. Over the next 18 months, we plan to do long-term testing and toxicology testing, and the aim is to have a clinical trial starting in 2022. So it's important to recognise that we know that this is only one gene out of the 62 retinal genes that we've identified in the ward study, so we still have a long way to go. So the take-home points of this presentation is that the bottleneck for developing a personalised drug therapy for a patient with an inherited retinal disease is not a lack of scientific knowledge, it's a lack of resources and funding. So if we have enough interest and funding, really 12 months is all it takes. The ward study is established in Perth, and it aims to fill the gap between a genetic diagnosis and a drug treatment trial. And the RP11 program is currently on track with local academic and industry partnership. And just to conclude, thank you for listening. And this is the slide shows a photo of our team at the Perkins and Lions Eye Institute. And we would like to acknowledge all the patients and families with inherited retinal diseases, as well as donations from the patients. Thank you. So now,
All right, thank you so much, Rachel. You might like to turn your cameras on again, and so that everybody can see you guys. And we'll ask some questions. My first question was actually for Rachel, just in relation to the ward study, Rachel. Well, perhaps I better wait till you're unmuted.
Sorry.
Lovely. I just wanted to ask you, how do you select patients for the ward study?
well, we select any patients that are keen to be involved and have an inherited retinal degeneration. So any patients that are able to come at 6 monthly appointments and ideally live in Perth, and it's not COVID, so even if they live interstate, if they were able to attend.
So it happens in Perth, but it could be somebody, anyone from Australia could be part of that, can join the study.
Yeah, so we have a few patients from interstate, but it's made it difficult at the moment because they're unable to travel with COVID. And obviously we need to do all the retinal imaging in the clinic. But
right,
if we weren't in COVID, any patient could participate if they have an inherited retinal disease.
Okay. And you mentioned that it's not so much the scientific capability that is missing, it's the funding. What would it cost me if I came to you and said, I want to do this work? How much am I going to have to pay?
I'll pass that one over to Dr. Fred Chen.
Can you hear me from there?
Can
You
you?
might need to come a bit closer.
So it's Fred here. Just an example for a new drug to be developed for the RP11 program. The capital required to generate the preclinical data is at least $50 million.
Oh my God.
And that will only allow us to make an application to the FDA, not even testing the drug in a human yet.
Wow. Okay.
In order to test in a human, you need to sit down to listen to this. It'll be about $200 million to do.
Okay.
Now that's obviously for a rigorous process. Now there is examples where this can be done much more cheaply, and that's in the States where they have developed a treatment for epilepsy called Batten disease, where one person received the drug, and I don't know how much, it's undisclosed how much money they've spent, but they set out a foundation. It's called the Millicent Foundation. And I can't imagine they would have $200 million to do this, but only one person had the drug and it's shown to be effective. And it's a purely personalized treatment.
So it's not something that the average man in the street can just roll up and ask for.
At the moment, no.
Okay, thank you very much. This is a more general question. What is the best process for individual patients to find out the cause of their specific IRD?
So I suppose there are a couple of different options. One is if you want to participate in the registry and our research, then we can enrol participants and analyse their DNA if their genetic diagnosis falls into one of our core projects at any given time. So it's really valuable because it's important that we continue to build this database of IRD affected Australians. So the other option is that you could, and it depends on a number of factors, I suppose, if you're family planning and how quickly you need a genetic diagnosis or would like a genetic diagnosis. You could go to your local genetic service, and it's possible then that they could arrange genetic diagnostic testing. And I think, again, it does take a little while, and you're not necessarily guaranteed to get genetic diagnostic testing, but that's also another option. And I have to say that at the registry, our testing is on a research basis, while obviously the quality is incredibly high. It still needs to be confirmed in an ARDA accredited laboratory, which Terri touched on. It's just the hard work would have been done. We would have found the genetic mutation or the genetic faults, which is a really difficult bit and the time-consuming bit, but they can be verified relatively easily.
And how long does it take to actually do the work on the DNA sample that you receive?
Again, it depends on a number of factors. It depends on their diagnosis, how many genes are implicated in the disease. So, for example, it's relatively easy, I suppose, to identify a genetic cause for disease that might be choroideremia, because there's only one gene and it's quite small, for retinoschisis. But for for diseases like retinitis pigmentosa, it can be really difficult. We first have to try and find the disease-causing mutations. Then we have to try and figure out which of all these genetic modifications that we've identified could actually be disease-causing. And sometimes we're not left with a really conclusive answer, so it could be inconclusive.
I just want to add something. It's actually critically important to get the phenotype correct, because if that's wrong, then you go nowhere. So in fact, it's probably the most important thing of all this process is to see the right specialist who can identify the retinal changes and to hypothesize these are the set of genes that need to be looked at. Because there's not much point, getting a genetic testing done when your question is, could this vision loss be due to inherited retinal disease? Because you've got to be much more specific than that. And unfortunately, the clinical geneticists who receive referrals for diagnosis often don't get enough information to work on. Some of the referrals say there's vision loss. Well, there could be hundreds of different causes of vision loss. And then in addition to that, you need to say retinal disease. What kind of retinal disease? Is it Stargardt's? Is it choroideremia? Is it this type of RP or that kind of RP? Are there any other changes on certain scans that suggest one gene rather than another gene? So there's a long process to get to that point, even before the clinical geneticist gets involved.
And could I also add to that, it's for this reason that at the registry, we use a non-hypothesis approach to testing. So we cast the net wide, we cover all bases, which can be good and bad, so even if we've got our clinical diagnosis wrong, for example, then we could still identify disease-causing genes and then go and verify with clinicians and so forth.
Okay, and what are the costs associated with coming and sending a sample to you for testing?
To the registry?
Yes.
Yeah, it's no cost. Yeah, it's funded. So there's no cost to the participant. We would enrol them. Lynn Hoffman, Isabella Camp, or Irwin now, talk to the participants and get as much information as we can about the phenotype and family history, and then we'll send out a blood or saliva, preferably blood, collection kit, and it's all at no charge to the participants. The collection, the analysis, and the reporting. The trade-off is that it doesn't happen quickly like a normal diagnostic test.
It's not actually true. It's free.
Oh, well.
If I could
yes, good.
put my 2 cents worth in, Sally. Ideally, you'd like to be able to go to a genetic service in your state and they would test for you. It would all be covered by Medicare and so on. Now, Medicare doesn't cover this at all. So it varies from state to state and institution to institution. So some people might say, I went to my local genetic service and I got a test done. And then someone in another state or for another reason can't get a test done. So we're sort of a de facto, we do research, but falling out of our research, we have produced 1,000 reports. But nevertheless, you could send us DNA and we might look at it for 10 years. We're not a clinical service, but we attempt to do our best to report whatever we find as part of our research.
And also, I should clarify, sorry, that it's not free. It's at no cost to the participant, but it's a great cost to us and our funding bodies.
Yeah. A typical test might be $1,000 to do the analysis, but then there might be another $2,000 of salaries of our salaries to interpret what's happened in that genetic analysis.
Yes.
So it's typically, you'd say $3,000 is a good round sum to produce from nothing to producing a report.
I think I've just got one. Oh no, there's another question here. Hold on. Oh, somebody was just asking whether we were recording the session. Yes, we are recording the session, and you'll be able to watch it again later. That's to all the people who've attended. It sounds so exciting hearing these stories about the gene therapies and all the new things that are, and certainly Luxturna, which is just about to arrive in Australia. I wonder if one of you would be prepared to imagine what we might be seeing maybe in the next 5 years and maybe in the next 10 years as where we might be for people with inherited retinal disease.
Fred might be in the best position to answer that as being the clinician.
It's not coming fast enough for me. What I envisage is that for any children or adults presenting with an inherited retinal disease, that there is a specialized center where they can present to get fast-tracked DNA analysis to get a diagnosis. And based on the genetic mutation or the gene that's involved, they will be offered a standard of care therapy, whether it's in the form of a drug that can be injected into the eye or gene therapy to repair the mutation. In 10 years' time, I anticipate there will be at least a dozen gene therapies that will be standard care that we can offer. In addition to that, the other ramification of this is that because of the easily accessible genetic diagnosis and counseling, there may be a reduction in the number of people born with inherited retinal disease because of family planning and technology have been so advanced to prevent more of these diseases from occurring in the community. So on two fronts, there will be less morbidity and suffering in people with the disease, and on the other hand, there will be less people born with these diseases.
It's certainly an exciting time, and we're delighted that you could all be here today and speak to us, and that you could also come along, Dr. Chen. So I just want to say on behalf of Retina Australia, thank you very much to all our presenters, and thank you to our attendees for coming, and I hope it wasn't too difficult to join and that you were able to gain a lot despite our technical difficulties. And we really do hope that we'll be able to run some more webinars in the future. When you leave this webinar, I think that you're going to be taken directly to a survey where you can give us some feedback about this session. And please feel free to write whatever you like and as much information as you can, because it will help us in planning for the future. So I think the final thing is just to say thank you very much, and until next time, all the very best.
Thanks, Ellen.
Thank you.
Thank you.